Hi, I would like to introduce myself. I'm Astri, the resident of pediatric surgery from Erlangga University. I would like to introduce about my research, the abstract of my research. The title was the wound healing of gaster perforation with human dried amniotic membrane as a biomaterial patch viewing from the Fibroblasts count and collagen density. Gaster perforation in neonates is a very rare case and have a really high mortality rate. It's about 75%. And with the condition of the neonates really have a high risk of the leakage of anastomosis, and there's a lot of surgery technique and material patch to prevent the leakage anastomosis. One of it is about the human dried amniotic membrane, which have a lot of advantage of wound healing. It have a lot of effect to have a better wound healing process, and that's why we want to prove that there was a difference in the number of fibroblasts count and the collagen density, to prove that in the case of gastric perforation, which have a patch of human amniotic membrane, have a better wound healing than the other group which doesn't have human amniotic dried membrane. We use New Zealand rabbit. We divided into 3 groups. The first group was primary repair in gastric perforation model, and the second group was about omental patch. We use a technique omental patch, modified Graham in the model of gastric perforation, and the last group is we used human amniotic membrane, dried membrane, to patch the gastric perforation in animal model. We used 30 rabbit, New Zealand rabbit. All the gender was male. We choose each group is 10 samples, and in our research there is no dropout. The samples there is no dropout samples, and from the examination results we found that significant difference of the fibroblast count for each group. We use Kruskal-Wallis test for the statistic, and then about the collagen density we find a difference, but in statistic in one way ANOVA we didn't found the significant difference, but we found the difference about the density is better in the group with human amniotic membrane. When we use a human dried amniotic membrane, then the other group have a lot of better wound healing process than the first group, the primary repair and the omental patch group. We found that there is an increased number of fibroblast count and the collagen density in the group with repair with human dried amniotic membrane in gastric perforation model in animal studies. Then the other group, which is not used the human dried amniotic membrane in repair the gastric perforation in an animal study. It's hopefully we can use human dried amniotic membrane for the future in human studies for this case, especially in gastric perforation, especially in neonates, or it's in children, which have a lot of risks for a leakage anastomosis. Hopefully it became one of the best choice method and biomaterial patch for healing process. This is my abstract of my research about human dried amniotic membrane, especially in perforation, gastric perforation in animal study. Thank you.